In the last video, we were talking about the plasma membrane or the cell membrane, and I said one of the most important functions of the cell membrane is that it acts as a gate that allows stuff to get in and out of the cell. So when we're talking about moving particles across a cell membrane, so moving stuff inside the cell to outside or outside to inside, we call that cell transport. Now, in this little video now, we'll talk a little bit about, we'll introduce this idea of cell transport. Now, in order for something to be transported or moved across the membrane has to be permeable to that substance. Now, permeability refers to the membrane's capacity or its ability to move a certain object or a certain substance from one side of the membrane to the other. So if a cell membrane is permeable to a substance, then it can move across the membrane. If the cell membrane is impermeable to a substance, means it can't move across the cell membrane. And then in most cases, our lipid membranes, since they're fluid, since they're made up of these protein channels and these interlocking lipids, they tend to be selectively permeable. Now what that means is it can regulate what gets in and what gets out. So depending on what the cell needs, it can find a way to get things in, and it also finds a way to get things out. So generally we'll find most cell membranes are selectively permeable to what it allows in and what it allows to escape. Now, the selective permeability of a membrane has a big role to play when we're talking about homeostasis. So depending on the condition of the cell, certain things can get in at different times, so on and so forth. So it's another way that the body helps regulate or maintain homeostasis. Now, a couple of things are going to affect the permeability of a membrane, and three things right here that we'll talk about now. We'll talk about the size of the molecule, we'll talk about the solubility or the solubility of the molecule in water, and we'll talk about the concentration or the electrochemical gradient of a substance. Now, these next couple of slides are pretty detailed and there's lots of information there. What I want you to take away from these next few slides is the big idea, okay? Not the little tiny details, but the big idea of each one. Alright, so let's go through them. Alright, so the first factor we'll talk about is solubility. Now, big thing about solubility is the molecule polar or nonpolar? And the reason why that has a big effect is because we're talking about two solvents. We're going to talk about the lipids that make up the membrane and water, which is kind of our universal solvent in our cells. So the membrane is mostly composed of nonpolar lipids, which makes nonpolar molecules highly permeable. So there's a rule in chemistry called like dissolves like. You'll hear about it in chemistry. So it's really easy for a nonpolar molecule to interact with another nonpolar molecule. So that increases the solubility. So it's more hydrophobic or lipid soluble, the greater the membrane's permeability to that substance. And I don't want, like I said, I don't want you to remember the big details. What I do want you to remember is these substances, in order to move across the cell membrane, they have to dissolve. They either have to dissolve in water or they had to be able to dissolve in lipids. Okay, so solubility, when we're talking about solubility and permeability, a substance can only move across the membrane if it's soluble in water, so it can dissolve, or soluble in lipids, so it can pass through the lipid membrane. Okay, big molecules like starch, for example, are insoluble. Then almost all membranes won't allow starch to pass through just by itself. So keep that in mind. It's not really worried about the nonpolar or anything like that. Is a molecule soluble in water? Is it soluble in lipids? And that will dictate whether or not it can pass across the plasma membrane. The next factor is going to be the size of the molecule. Okay. Now, generally, the smaller the molecule is, the easier it is to pass through a plasma membrane. Once we get into really big molecules like polysaccharides, polypeptides, proteins, these molecules are quite large and it's very difficult for them to pass through. I like to, when I'm talking about the size of a molecule, I always talk about the example of a fishing net. everyone understands how a fishing net works, right? You put out a net, it's got the different holes in it. So think about our net as our plasma membrane. And our plasma membrane will have pores in it. It won't be like a fishing net, but it will have little places where things can get through relatively easy. If you have a certain size hole in your net, then the little fish will be able to pass through no problem. Okay, so just like the little molecules, they'll be able to fit through these holes and they'll go on through the membrane. Now, when we get to the bigger fish, and I'll apologize because I am an awful artist, the holes in the net are going to be too small, so those big fish aren't going to be able to fit through. And that's just like a big molecule. So if you think about these little fish going through the fish net, these are going to be, your oxygen, your carbon dioxide, glycerol, steroids, little molecules that are easily pass through that net. Once we start talking about the bigger fish, now we're talking about the polysaccharides and the polypeptides. They're going to be too big, so they won't fit. So that'll affect the way that the cell moves them across the membrane. And keep in mind what I said, I'm more worried about you guys understanding the bigger picture of these ideas than the little tiny details. And finally, we talk about the concentration or the electrochemical gradient. A lot of times in a normal situation of a cell, there's going to be differences in the charge, electrochemical charge on the outside and on the inside of the membrane. And that's because we have a buildup of ions which have a negative and positive charge, and those charges kind of need to be in balance in order for us to be in homeostasis. One thing we need to remember is that a lipid membrane is impermeable to ions. Okay, so if you're a charged chemical species like an ion, you will not be able to pass through a lipid membrane. So what ions do is they use these proteins that are found in the membrane. You can see one example is right here. I think it's gramicidin. That'll allow charged molecules, in this case like chlorine or sodium, to pass through the membrane, because it's not passing directly through the lipids right here. It's going through a hole in the membrane caused by this protein. So ion balance between the inside cell of an outside of the cell, especially in our nervous system, is super, super important. Okay, so how the cell's plasma membrane interact with those proteins, how many those proteins are present in the membrane, will make a big difference on how we can move ions across the cell membrane. Now, those are factors that will affect how a membrane reacts to a substance. Okay, its solubility, its charge, and the gradient of the charge. So how high is the concentration on one side of the membrane compared to the other, and the size of the molecule. The next thing we have are two different forms of transport, two different ways we can move things across the cell. The first one we have is a passive transport. Okay, a passive transport is when a molecule will be able to move across the cell membrane, but it won't cost us any energy. We don't have to put any effort into it whatsoever. Okay, so think about passive transport as rolling down a hill. Okay, we can move an object by rolling it down a hill, and it doesn't cost us any energy. Just natural forces take it, in this case gravity, and it pulls it down there for us, and we don't have to spend any energy whatsoever. And the way that works is that molecules will move down their concentration gradient. So if there's a high concentration of a molecule on one side of the membrane and a low concentration on the other side, well, as long as that substance is the membrane's permeable to that substance, then those molecules will move from the high concentration area to the low concentration until we get an equilibrium, because nature hates everything to be out of equilibrium. And that's a form of passive transport. It doesn't cost us any energy, but we still manage to get things moved around. So if you look at the diagram, you can see there's two examples here of passive transport. We've got diffusion, and here's our concentration gradient. So this molecule right here, this little red circle, this is permeable. The membrane's permeable to this. We've got a high concentration on this side. We've got a low concentration on this side. So the molecules will move back and forth until we're equal on both sides of the membrane. Okay, and that's an example of diffusion. Another example is facilitated diffusion. I know facilitated diffusion works in the same principles, except think about these little triangles as some type of an ion. So it's got a positive or a negative charge. It can't pass through the membrane. Okay, so the membrane's impermeable, but it uses one of these protein channels, like the garamycin that we've seen on the other slide, and it'll move freely back and forth through this channel until we'll get an equilibrium on both sides. The second type of transport is active transport, and we've got an example down here. But active transport is when we need to have energy involved. Okay, so if we want to move something, we have to put some energy in it. So you think of active transport is if we need to get something from the bottom of a hill to the top of the hill. Well, instead of just letting it roll down the hill like passive transport where it doesn't cost us any energy, in this case we've got to get behind it and we've got to push it up the hill and we've got to spend some energy. So when we're doing this, we're actually moving things against its concentration gradient. So we're going from an area where we've got a small number of particles. So here's our membrane right here. Here's our small number of particles, and then we're going to an area where we've got lots and lots and lots of particles. So nature wants to move this way so that we get an equilibrium. With active transport, we want to move in the opposite direction. So it's going to cost us energy. We're going to take the concentration from the smaller area, and we're going to move it into the area of larger concentration. So here we're looking at hydrogen ions. We're going to use a protein channel because it's an ion, and it's going to be pumped in here, and it's going to go from an area of low concentration to an area of high concentration. Okay, and that's active transport. It costs its energy. We have to put energy into it in order for it to work.